The overall goal of this procedure is to fabricate a DNA origami nanorobot. This is accomplished by first preparing the staples pool and reaction mixture. Next, a thermocycler is used to gradually lower the mixture temperature to facilitate annealing of the short staple strands to the long circular scaffold. Then, the nanorobots are purified from excess staples using centrifugal filters. Finally, proper folding and purification are verified first by agarose gel electrophoresis. Ultimately, transmission electron microscopy is used to show the correct architecture of the DNA nanorobot shape.
Hello, I'm Yaniv Amir from Bachelet's lab, and I will be demonstrating this procedure.
Begin with DNA nanorobot staples in 96-well plates, lyophilized and normalized to 10 nanomoles. For a detailed description of the design and architecture of the DNA nanorobot, refer to the following references in the text protocol. Using a multi-channel pipetter, reconstitute the 254 staples by adding 100 microliters of RNase-free ultra-pure water to each well, to a final concentration of 100 micromolar. Then, using a multi-channel pipetter and a sterile 55 mm solution basin, pool together 20 microliters of each staple. For a standard reaction mixture, start with 40 microliters of M13-MP18 viral genome circular single-strand DNA, corresponding to 4 picomoles of scaffold DNA, or a final concentration of scaffold in the fabrication mixture of 20 nanomolar. Next, add 102 microliters of staples to reach a scaffold-to-staples ratio of 1 to 10, respectively. Add 0.4 microliters of each of 4